Welcome from my side. It's a pleasure to give this last talk, and I try to be on time, considering that I'm a little late already. And today I'm going to talk about correlation of transport of substances into cells and model systems like vesicles with shear flow and membrane state. An alternative title could be an example how state can regulate function. And as Matthias said, I'm a former PhD student of Matthias, and there I picked up the idea how state and function can correlate, or how state can lead to a function, and we are interested in how this state can be controlled. There we study different aspects like transport, enzyme activity, cellular interaction, and higher functions of cell ensembles. And we use some specific tools, surface acoustic waves, for dynamic force fields to tune cells or vesicles or microparticles. About these topics, we presented some posters already, and today I'm going to talk about nanoparticle and drug transport in vesicles and cells. So from an application side of view, the basic idea here is if we can make use of increased permeability of substances that do not like to go into cells, close to a phase transition, to identify potential new drugs. And therefore we study in vitro cells, and we envision somehow some state diagrams of uptake, like similar as we have got shown by FIF yesterday for excitability, for uptake into cells. And the axis could be, for example, temperature and shear, or shear rate in a parabolic flow profile and elongational flow in a microfluidic setup. And yeah, we want to identify such phase diagrams and then finally exploit those phase diagrams. Measured ones could, for example, look like this. And then we want to go to these points, switch the permeability of cells, and screen compound libraries together with pharmacists to identify potential new drugs that then could get some shell or some transporter to get them into the cells more easily. And as a kind of calibration system, we do the same stuff with model membranes. And then to extend our parameter space, we apply surface acoustic waves to maybe further enhance the transport. Let me start with nanoparticle uptake in vesicles. Some years ago we started to measure uptake of silica nanoparticles in cells in vitro together with Matthias and Matthias' brother. And we saw also that the biological response scales with the total surface area concentration of the nanoparticles. And with this knowledge, we also studied then the interaction of these nanoparticles with model membranes, as you can see indicated here. And a typical experiment then looks like this. You have nanoparticles distributed all around, and with increasing time, the nanoparticles are taken up by these lipid vesicles in a way that they adhere to the membrane. The membrane is wrapped around the particle, and then finally, when this membrane neck breaks, water can flow through this pore to release the membrane tension that appears by the nanoparticle taking away surface area from the vesicle. We studied this from a mechanical point of view and measured adhesion energies between this material and lipid membranes, and for example could show that the adhesion energy as function of salt concentration can be switched from non-adherent to adherent at a critical salt concentration. And we find the same behavior in uptake of nanoparticles in these vesicles, and this critical salt concentration turned out to be size dependent of the particles. What makes sense if we, by this adhesion energy here, could tune the ratio of adhesion energy and necessary bending energy, and energy to overcome the membrane tension to form the pores. But these mechanic parameters also have to be thermodynamic parameters, and thus it's not surprising that the uptake rate as function of membrane state, or measured for different temperatures, for lipids with a first order phase transition, show peaks or maxima at distinct temperatures. Here for 15 pC vesicles you can find maximal uptake very close to the phase transition and differences in uptake rate in the fluid phase and in the GL phase. And if we now slightly change the lipid and make it a little shorter, use the MPC lipid, we find pronounced uptake maxima close to the phase transition of these lipid membranes. And of course, in turn, the adhesion of the nanoparticle can influence the membrane state, as we have shown in my PhD thesis, for example. And this can be understood then that the nanoparticle could itself change the state of the membrane, make it more permeable, and enter the vesicle, or maybe also the cell membrane. That vesicles or lipid vesicles are permeable close to phase transition has been shown around 10 years ago by Thomas, using dye molecules and measuring the leakage of the molecules of the vesicles. And you see a very nice correlation of heat capacity, what is a measure for the phase transition, and the permeability of these vesicles. We did here something similar, but we used larger molecules with sugar groups that typically stay within these vesicles, and we sent the filled vesicles then through a tube in the range of some 5 times the vesicle size, control with the heat bath the state, like the membrane state, and then the flow rate, and measure how much of the dye is released from these model cells. A typical micrograph then looks like this. You see in red state the membrane, and in green the molecules we put into these vesicles, and what we found is a all or nothing behavior in kind of release. Either the vesicles are empty or they are full. And you can also quantify this by measuring the overall intensity within a vesicle. And so we decided to take as a measure, like counting or measuring the intensity of filled vesicles and empty vesicles, and repeat this experiment at different temperatures and different flow rates. So here you see the amount of full vesicles as function of temperature for one distinct flow rate. And you see the heat capacity profile indicating here the transition of the used lipid membrane. You see this nice correlation that in the GL phase we have no release of the substance, and in the fluid phase we have a distinct amount of vesicles that could release the substance. If we now increase the shear force, we find that even more vesicles are empty in the fluid phase, but also around the transition here in the GL phase, vesicles can release cargo. And if we further increase the shear rate and thus the shear force, we find an even stronger behavior. Now, to interpret this trend, we just started measurements as shown by Simon yesterday. Like what we now are gonna do is measure the impact of shear force on membrane state optically to answer the question whether this effect can be understood like this. Shear force influences membrane state, like for example a shift in transition or broadening of the transition, and that this then leads to the increased permeability, or if it's more a tiny effect of the shear force on the state diagram, and here we simply overcome the compressibility of the GL phase by higher forces. So yeah, unfortunately I cannot answer this question, but I can show the phenomenon here. But even if we cannot really correlate now the membrane phase transition and these transitions in the effect, we can try to see the effect also in living cells. And that's what we did here. Like our hypothesis now would be, if we take cells and go somewhere here in a state diagram, for example at room temperature, if we would have no shear force, we would expect somewhere around the transition there should be high uptake into the cells, and above maybe some uptake and below a low amount of uptake. If we now increase shear force, we go through this diagram on the shear axis. And we realize this by bringing together in a wide junction, in a microchannel, cells and the drug. And then we mix it here in a tube, and we do this for different shear rates. What you can see here is, this is the nucleus in blue, and here in green you see the FITC-Dextran, the molecules that do not pass the cell membrane easily. And we find uptake into the cytosol and also in some kind of vesicular structures. So now we go along this axis and increase the shear rate. And that's what we find. We find an increase in the uptake of these molecules into the cells, but not just a linear increase, but a maximum at some distinct flow rate. So this is compatible with the interpretation that the shear force may regulate the membrane state, that may regulate the permeability. And we are going through the state diagram like this. And of course, if it will turn out that it's actually the membrane state that is influenced by the shear force, then of course we could use other parameters, like not only shear, but temperature, pH, calcium ions, or other thermodynamic forces to shift our state diagram, or to find a point in this multi-dimensional state diagram to really have increased transport into the cells. Now, if this is related with the membrane state, then it should be reversible. As it's not so easy now to measure this transport with the identical cell several times and go across the transition, we designed the experiment to somehow prove the reversibility. And what we did is we treated all cells with a very high shear rate, then aliquoted our sample, and this we did in absence of the drug, and afterwards we used these aliquotes to treat the cells with the drug in the same concentration but at the different flow rates again, and we could reproduce that we find an increased uptake at the flow rate and shear force we had before. So this kind of reversibility of our permeabilization of the cells is consistent with this argumentation, with this hypothesis. And now from an application side of view, it could be interesting to even further increase the permeability of the cells, and that's why we use surface acoustic wave chips as tools to additionally manipulate the cells. Due to time reasons, I will skip how you can create these waves, but it has been demonstrated very nicely what you can do with a wave with a 1 nanometer amplitude roughly when it is in contact with aqueous solutions. You can, for example, use it to stir and mix in droplets or microchannels. You can actuate droplets, microliter sized or smaller, on a chip to, for example, fuse droplets and get chemical reactions started. And you can also manipulate particles in standing wave fields, as indicated here. We create waves from here and here and here and there, and by this we get a standing wave pattern that we can manipulate to get a distinct pattern, for example, of particles or also living cells. And what we now do is we combine somehow this particle trapping and mixing behavior, and we set such a shear, a microfluidic channel on a saw chip to additionally apply a surface acoustic wave to mix our cell solution and the drug solution, and then trap the cells additionally here in this vortex. What then looks like this, and here you maybe can guess the trajectories of cells caught here in this vortex. They travel on closed trajectories in this vortex while the substance containing the drug here is flowing across the trapped cells, and later on we analyze the uptake of the substance then into the cells. What we find is a very narrow parameter window that can be tuned by tuning flow rate and applied power of the acoustic wave. We find an optimal treatment time of the cells in this trap for about 2 minutes, and this is the summarizing graph. We here show that this is the reference static incubation of the cells with the solution of the same concentration. Then if we add flow, we can increase the uptake by a factor of about 10. If we additionally use this saw trap, we can increase it by a factor of about 60. And if we now go to larger molecules like FITC-Dextran, we can still have an increase of about a factor of 10 compared to static incubation, and this also works for proteins like GFP. And so currently we also did some experiments with CRISPR-Cas to maybe get some biological effect in the cell, but yeah, they are ongoing and yeah, but maybe we can tell about this in some months. Okay, at this point I want to thank my mentors and my dynamic group and you for your kind attention, and now I'm looking forward to questions.
Thank you very much, Christoph, and yeah, he's happy to take some questions. Anybody?
Martha, can you just be loud?
Yes, I really enjoyed the talk, and I just the question of phase transitions in cells is particularly of interest to some of the people here, but still, just from the fact that you do some experiments and get these nonlinear things, just from the fact that you make the measurements on living cells and you get some nonlinear response on the uptake, how can you add more information with your capabilities into what is going on? Because still there will be...
Yeah, still, like, this is what I tried to say in the beginning. The original plan would be to not only measure the phenomenon of uptake as function of membrane state or in a parameter space, but also measure membrane phase transitions optically. Like, this is ongoing, so we have not done it successfully yet. So,
okay.
But this is our plan then, to correlate both.
And to answer the world outside of this room, are there ideas related to protein transporting mechanisms that take in these fluorescent particles you're using? Or?
Yeah, so we have indications that for the living cells we have uptake directly in the cytosol, so these may be pores or should be pores in the membrane, and we have these kind of vesicular structures where proteins may be involved, and we all know that proteins also can react on shear forces and unfold or change the conformation under shear force, so I do not doubt that these proteins, membrane proteins, also contribute to the function. And like I would doubt that we will find a strict correlation of some model membrane parameters phase diagram and the phenomenon, as also other forces involved scale on these axes. For example, if we study the nanoparticle uptake and we go on the pH axis, the surface potential of the nanoparticles also changes, and this can also change the adhesion energy, and it's not only the membrane that is state dependent. Yeah?
Do you vary frequency of your saw? I mean, do you use effects that your saw has a specific wavelength in respect to the size of your cell to look for some resonant phenomena?
Like here we choose the frequency and thus the wavelengths to get trapping of single cells, or at least to get trapping of cells, and in some cases we try to get single cells per well in the potential. And we also, in some cell stimulation experiments, we plan to vary the frequency to meet maybe some mechanical resonance of the cytoskeleton or the combination of cytoskeleton and membrane to somehow bring the cell in resonance. But yeah, that's ongoing. Aki, you had a question?
Okay, so be loud. So first of all, thanks for the very nice talk. Unfortunately, your computer is already plugged off, but in one of your first slides about the nanoparticle uptake, you showed a graph that indicated that in the gel phase, of course, I mean, uptake in phase transition is highest, but then if you compare gel and fluid phase, you had a higher uptake in the gel phase compared to the fluid phase. Do you have an explanation for that? I
Um,
would have expected from a first guess that it might be the other way.
Yeah, I would have expected also the other way, and but maybe it's not only the compressibility that to open up pores, but also that the bending energy is much higher in the gel phase, and to favor the breaking of the membrane, of the neck of the wrapped particle close to the membrane, so that this makes it energetically more favorable to happen in the gel phase than in the fluid phase. And yeah.
Maybe one last question, Carlos?
So, if you lower the pH, do you think it's going to be different?